know, even your lab glassware holds many microorganisms which are very dreadful to your eyes? So how do we make them micro-free before starting any experiment? Let's decode the science behind the topic called as sterilization. Hello everyone, welcome back to my channel Genius Minds. In today's video, we are going to talk about very important topic which is known as sterilization. So let's start it. It is defined as complete removal or destruction of the microorganism. So there should not be presence of a single microorganism in any of the glassware or any lab instrument whichever you are going to use of microorganisms from the environment. This is proper definition of sterilization. Now when we talk about this microorganism, it can be bacteria, it can be fungal spore, also it can be any form of viruses. So it should be contamination free. Not even a single colony or species should be existing there. Now, how do we perform this sterilization? There are different ways how we can perform sterilization. So let's have a classification of the different methods. So sterilization, broadly it is divided into two categories, that is physical and chemical. Physical methods of sterilization, chemical methods of sterilization. Let's talk about the physical methods of sterilization first. The physical method of sterilization is again divided into two categories first, heat and radiation. The heating method, again we are having two methods over here, that is dry heat sterilization and moist heat sterilization. When we talk about the dry heat sterilization, mostly in the labs we use an instrument which is known as hot air oven. And when we talk about moist, we use autoclave as an instrument as a moist sterilization method. Radiation involves the use of both the method, that is we are going to make use of ionizing radiation as well as non-ionizing radiation. So when we talk about ionizing radiation, we prefer using gamma rays as well as X-rays. And when we talk about non-ionizing radiation, mostly at most of the place, UV rays plays an important role over there. So these are all the physical method of sterilization. Now when we talk about the chemical method of sterilization, it is again classified into two important categories, gaseous method and liquid method. So in the gaseous method, we are going to make use of gaseous agent. So mostly we use formaldehyde and ethylene oxide as a gaseous agent to sterilize any object. When we talk about the liquids, which are mostly used even in our day-to-day life and in the lab especially, the chemicals what we are using here are alcohols, aldehyde, phenols and phenolic compounds as well as halogens. So these are the liquids that we are using in our daily life or in a routine lab experiment to sterilize our laboratory area and to make it micro-free or free from contamination. So that's all for today's video. If you are new to my channel, please do subscribe it and please share it. And if you have any doubt, you can drop a comment. And if you want videos in detail about each one of them, please do drop a comment. Thank you so much.